Hello everyone. Today's topic is application of biotechnology in medicine and health. As we know, biotechnology has revolutionized modern medicine from curing genetic diseases to creating life-saving drugs. This powerful science is shaping the future of healthcare. Whether you are preparing for UPSC or NEET examination, understanding how biotechnology is applied in medicine is essential. It is not just for exams, it is for grasping how science is transforming lives around us. In this video, we will explore key medical application of biotechnology, how to use gene therapy, application in field of vaccine production, production of therapeutic proteins, production in personalized medicine, different types of diagnostic tools, human insulin production. So I will explain simply and clearly. So let's dive in. Biotechnology in India, about biotechnology in India. In India, the Department of Biotechnology under the Ministry of Science and Technology plays a pivotal role, plays an important role in managing biotechnology programs. Objectives include research, infrastructure development, human resource generation, biotechnology popularization, industrial promotion, and biosafety implementation. The Genetic Engineering Appraisal Committee GEAC, operating under the Ministry of Environment, Forest and Climate Change, evaluates activities involving hazardous microorganisms and recombinants. India's biotechnology sector has experienced extraordinary growth. It is showing skyrocketing growth, skyrocketing from $10 billion industry in 2014 to over $130 billion in 2024, with projection to reach $300 billion by the year 2030. India is not only a leader in biotechnology, but is now all the center of a global biotech boom, which will foster innovation, create jobs. Different types of job is creating with the help of biotechnology. It is strengthening environmental commitment. So biotechnological application in medicine have revolutionized healthcare by providing advanced solutions such as genetically engineered insulin, gene therapy, gene silencing. This innovation not only improve disease treatment and management, but it also offer sustainable approaches in bioenergy and environmental biotechnology. So India currently accounts for 60% of global vaccine production. India currently accounts for 60% of global vaccine production and is home to the second largest number of US FDA approved manufacturing plants outside the United States. With growing investment opportunities across sectors such as the biopharma, bioagri, bioindustrial, bioenergy, bioservices, Medtech India is emerging as a global leader in biotechnology. So India is currently accounts for 60% of global vaccine production, which is second largest number of USFDA approved manufacturing plants outside the United States. The ongoing bio-revolution in India is the IT revolution driven by the West, highlighting that the country's rich natural and biodiversity resources are key to its biotechnology success. It also praised the efforts of Indian startup. It also praised the efforts of Indian startups leveraging biotechnology to create innovative solutions such as non-human milk and other sustainable products. The Union Minister recalled the launch of the Mission Suraksha initiative, which led to the development of indigenous DNA-based vaccines and world largest vaccination drive during the COVID-19 pandemic. In the year 2024, India's biotechnology sector has also seen a notable, remarkable achievements, such as the development of the world's first human papillomavirus vaccine against the cervical cancer and the breakthrough creation of the indigenous antibiotic nephithromycin, which has proven effective in treating respiratory diseases. Furthermore, the Department of Biotechnology has successfully developed a gene therapy experiment for hemophilia. Hemophilia is a disease which when a person get injury and the blood will not clot, it will continuously bleed. In the past, India would often take cues from other countries in terms of technology and innovation. Today times have changed. Other countries are now looking to India for guidance and inspiration. So how the India is developing in the biotechnology field. India is proud to be leading the way, particularly in areas such as quantum technology, where the country is making significant development, significant strides with the quantum mission. India's remarkable advancement in biotechnology and other emerging technologies position it as a key player on the global stage, driving the future of innovation, sustainability, and economic growth. Biotechnology has revolutionized medicine, offering new approaches to diagnosing, treating, and preventing diseases. It is driven by advancement in molecular biology, genetics, nanotechnology, leading to innovative treatments for conditions like cancer and genetic disorder, as well as helping the production of different types of vaccine and diagnostic tools. This field is very important, very crucial in responding to emerging health threats, as seen during the COVID-19 pandemic. We have encountered so many health threats during the COVID-19 pandemics. Here is more detailed look advancement and application in the field of biotechnology. It gives us different types of therapeutic proteins, which it has enabled the production of therapeutic proteins like recombinant insulin for the cure of diabetes, offering a safer and more reliable alternative to traditional sources. It helps in production of CRISPR technology, for example, allows for precise editing of the genome, holding promise for treating diseases. We are able to treat the hereditary diseases, genetic diseases with the help of gene therapy methodology. With the help of biotechnology, we are developing different types of vaccines for infectious diseases like HIV, which is HIV is a reason for AIDS, and the recent COVID-19 vaccines. It also led to the development of advanced diagnostic tool for various diseases, including infectious diseases. There are some challenges and consideration in the field of biotechnology: ethics, cost, regulation, accessibility. Ethics, a biotechnology advances, it raises ethical consideration regarding genetic editing, gene therapy, and the use of new technologies. Biotechnology products are little bit costly and more costly like biopharmaceutical products. It can be expensive, impacting access and affordability. The rapid pace of advancement in the field of biotechnology requires careful consideration, careful regulation of biotechnology to ensure safety and efficacy. For the biotechnology, it is ensuring that the benefits of biotechnology are accessible to all regardless of socioeconomic status or geographical location, is a key challenge. So these are the challenges in the field of biotechnology. What about the future trends in the field of biotechnology? It gives continued advancement in the field of gene therapy. Research and development in gene therapy are expected to lead to more effective treatments for a different types of genetic disease. It helps in the personalized medicine. The use of genomics and other omics technology will continue to drive personalized medicine, allowing for tailored treatment based on individual patient characteristics. So we are able to treat the patient individually. So we are giving the personalized medicine with the help of use of genomics and other omics technology. With the help of artificial intelligence and big data, we will play an increasingly important role in drug discovery, development, and personalized treatment plans. It helps in the regenerating medicine. It holds a promise for repairing damaged tissue and organs, potentially leading to treatment for conditions like spinal cord injuries and organ failure. So, biotechnology help in the treatment of the patient if the patient will get spinal cord injuries, for the it's also a boon for the development of different types of organ. So it is a treatment for organ failure also with the help of biotechnology. Other benefits are cell and gene therapies. These emerging therapies, like the first CRISPR gene therapy, Casgevy, offered the potential to revolutionize patient lives. Monoclonal antibodies has enabled the development of monoclonal antibodies, which also used to treat the different types of diseases. Nanotechnology, which is used with the help of microscope equipment that can enter the human body, opening new avenues for diagnostic and diagnosis and therapeutics. What are the impact on healthcare on in the for the biotechnology? It improved patient outcomes. Biotechnology has led to breakthroughs in treating and preventing diseases, improving patients' outcomes and quality of life. It gives personalized treatment. It faster discovery. With the help of biotechnology, it helps in accelerating the discovery and development of new therapies by leveraging genome targets and genetic information. The vaccine given by the biotechnology with the help of biotechnology are safer. It allows for the development of safer and more effective vaccines, including those that can be engineered to carry multiple strains of pathogens. These are the few applications of biotechnology. Applications are it uses in medical diagnosis to diagnose different types of diseases. It used in environmental and energy management. It is used in plant agriculture and crop improvements. It helps in the field of recombinant DNA technology, genetic engineering technology. It also used in the fermentation technology. It improves food technology. It also helps in healthcare pharmaceuticals. It also helps in animal breeding and agriculture. So, example of recombinant DNA technology in the health sectors, the first commercial healthcare product derived from recombinant DNA technology, genetic engineering technology, was human insulin. Today, it is successfully applied to make new antibiotic vaccines. Vaccine we have generated, that is the hepatitis B vaccine, and different protein products production system for instance, for insulin and for human growth hormone. So here are few example of recombinant DNA technology in the health sectors. With the help of biotechnology, we have given human growth hormone, which help in the treating growth disorder in children. See, with the help of human growth hormone, we are treating children who is having growth disorders. Erythropoietin. It also give rise to erythropoietin, which helps to manage the patient who is having kidney problems, kidney failures, and cancer, and the patient is suffering from anemia. So the treatment is given with the help of erythropoietin treatment. Other one is the tissue plasminogen activator. Recombinant DNA drive tissue plasminogen activators is used to dissolve blood clots to in stroke patients. If the patient will get stroke, with the help of tissue plasminogen activator, it is used to dissolve their blood clots. So it is a very useful advantage of biotechnology. Herceptin, that is a trastuzumab. This recombinant DNA made antibody targets HER2 positive breast cancer cells. So it is also it's very advantageous in the treatment of breast cancer with the help of the recombinant DNA technology, which made antibody targets HER2 positives, herceptin, trastuzumab. Factor 8, it's also helping the development of recombinant factor 8, which treats patient with hemophilia A and promoting blood clotting. So biotechnology application in medicine, have transformed diseases. It's help in the prevention, diagnosis, and management of different types of diseases, enhancing both the quality and longevity of human life. So with the help of biotechnology, the quality of human life, the life of human life is increasing. This can be broadly categorized in the following aspects. It's help in the development of drugs, different types of drugs. The drugs are pharmaceuticals, genomics, high throughout screenings, and bioinformatics. Biopharmaceuticals, it enables the production of therapeutic proteins, antibodies, enzymes, vaccine, and other drugs from biological sources like cells and microorganisms instead of chemical synthesis. This biopharmaceuticals transforming the treatment of diseases like cancer, autoimmune disorder. Disorder is a disorder in which self is destroying their own self. So it's a help in the treatment of cancer, autoimmune disorder like rheumatoid arthritis, infection, etc. It also help in the development of pharmacogenomics. It involves studying how genetic differences affect drug responses. It enables the development of personalized medicine tailored to an individual's genetic makeup. So we are treating the individually with the help of individual's genetic makeup with the help of biotechnology. And the field is a pharmacogenomics. Pharmacogenomics promises to make therapies safer and more effective, more efficient. With the biotechnology, there is a one more other development is the high throughput screening. These tools allow rapid screening of thousands of drug candidates against target diseases or infection to identify potential leads for further development. So this makes the drug discovery process quicker and more efficient. So it helps into the discovery the drug very easily and very efficiently. It also helps in the field of bioinformatics. It analyzes bioinformatics is the field in which we analyze the biological data using computer tool. It helps identify new drug targets and disease biomarkers. So bioinformatics is critical for drug discovery and development is post-genomic era. Bioinformatics is a multidisciplinary field that use computational tools that and methods to analyze and interpret biological data, particularly DNA and protein sequences. It combines computer science, biology, mathematics, and statistics to take complex biological questions. It helps in analyzing and integrating large biological data set using the specialized software tools and databases used across biomedical R&D, omics technology, clinical diagnostic, public health surveillance, etc. So bioinformatics is a very important field nowadays for the career also, for the engineering student also. Key aspects of bioinformatics itself in the data management, data analysis, modeling and simulation, interdisciplinary natures. It deals with the storage, retrieval, organization of large amounts of biological data, often in database. It utilizes computational method to analyze biological data, including sequence alignment, phylogenetic analysis, and protein structure prediction. It develops model and simulation to understand biological processes such as protein folding, gene regulation. It involves collaboration between biologists, computer scientists, mathematics, and other specialists. So example of bioinformatic application is sequence alignments, comparing DNA or protein sequences to identify similarities and differences. It helps in the phylogenetic analysis. Phylogenetic terms means to know about the heredity, to know about the evolution. So constructing evolutionary trees to understand the relationship between different species. It helps in the protein structure prediction. It helps in the gene expression analysis, expression levels of genes to understand their activity. It helps in the data mining, so discovering new pattern and insight from large biological data sets. Application includes gene sequencing analysis, molecular modeling, structure-based drug design, analysis of molecular interaction, disease biomarker identification. So its significance is a lot. It enables extracting knowledge and insight from the wealth of data generated through modern biological techniques, and it is a totally based on computational tools. Diagnostics, there is a different types of diagnosis developed with the help of the biotechnology, molecular diagnostics techniques like PCR, microarrays, next generation sequencing enable fast, sensitive, and precise diagnosis of infection, cancer, different type of genetic diseases, hereditary disorder by detection of biomarkers in patient samples. So companion diagnostic is there, which identify biomarkers to determine which patients are most likely to respond to a particular therapy, which patient to get the which therapy, different therapy. So it's based on the individual. They enable targeted personalized therapies. Biosensor, the other type of diagnostic tool is a biosensor. Biosensor incorporate biological molecules like enzyme or antibodies, can detect biomarkers rapidly and accurately. Biosensor have application in point of care diagnostic. Lab on a chip, it is also one diagnostic tool which help to microfluidic device integrate multiple lab processes from sample preparation to analyze on a single chip. They allow the cost is low, automated diagnostic at the patient beside or in the field. So there is a image is given about this is about the biosensor which is attached in the body, and second image is lab on a chip technology. Now, next one is the development in the field of biotechnology is the tissue engineering stem cell. Embryonic or stem cell can be guided to develop into various cell types. So, stem cell are able to generate different types of tissue. They have application in regenerative medicine and tissue repair. Biocompatible synthetic polymers or biological molecules like collagen are used as a scaffold for tissue regeneration and guiding cell growth. So we are using the biocompatible synthetic polymers or biological molecules like collagen for this purpose. Other tissue engineering stem cell is a 3D bioprinting. Cells and biomaterials can be precisely organized in 3D pattern to bioprint transplantable tissue and organs. This emerging field aims to address organ shortage. So it helps to develop the different types of organ artificially. The image is given 3D bioprinting materials techniques in situ one is 3D printed. This is the image is given about 3D bioprinting. It holds significant promise for addressing when the organ there is a shortage of organs. So 3D bioprinting plays a very important role for creating custom-made functional organ for transplantation. So it plays a very important role for developing a organ if there is a organ shortage. So it develops a functional organ for transplantation. This technology involves using a specialized printer to layer by layer build complex biological structures, including cell biomaterials, bioactive molecules, by mimicking natural tissue architecture. So 3D bioprinting offers a potential solution for creating organs that can be tailored to individual patients' needs, potentially minimizing rejection and improving transplantation outcomes. So it minimizes the rejection method during the transplantation and improve transplantation outcomes. Potential benefits of 3D bioprinting for organ shortage. Patient-specific organ, it allows for the creation of organs tailored to individual patients, to individual needs, reducing the risk of rejection, minimizing the risk of rejection of organ transplantation, and the need for immunosuppressant drugs. Immunosuppressant drugs is used, that time cyclosporine like that, to reduce the rejection. Organ transplantation. Elimination of donor dependence. By fabricating organs in a lab, 3D bioprinting could potentially elevate the reliance on organ donors, addressing the growing shortage of available organs. It improved transplantation outcomes. So better long-term survival and function compared to traditional organ transplantation. It can be used to create various tissue and organs, various tissue and organs, paving the way for regenerative medicine and tissue engineering applications. So 3D bioprinting plays a very important role for addressing organ shortage by creating custom-made functional organs for transplantation. What are the challenges in 3D bioprinting for organ transplantation? Despite the promise and advancement in 3D bioprinting, there are several challenges. Challenges are cell viability and functionality, scalability and complexity, vascularization and blood supply, ethical consideration. Ensuring the cell maintain their viability and functionality during the printing and culturing process is crucial. The cell should be viable, should be livable, and should be functional when we are doing the printing and culturing process. Second one is the scalability and complexity. Creating large and more complex organ requires further advancement. So there is a need of the advancement in printing technology and material science. This is a challenge again. Vascularization and blood supply. When we are doing this 3D bioprinting technology, there is a need of a functional vascular network within the 3D printed organ, which is essential for delivering oxygen and different types of nutrients. Then again, there is an ethical consideration. The use of 3D bioprinting, it raises an ethical question regarding organ sourcing, safety, and potential misuse. So, 3D bioprinting offers a revolutionary approach to addressing organ shortage and transforming the field of transplantation. While challenges remain, ongoing research and advancement are pushing the boundaries of this technology, paving the way for a future where personalized lab-grown organs become a reality. Now, gene and cell therapy. Other advancement in the field of biotechnology are the gene therapy, cell therapy, CART cell therapy, the CRISPR Cas system. So, gene therapy is correcting diseases causing which are the causing genetic defects by inserting healthy genes using a vectors with the help of vectors or other delivery methods. So gene therapy products have been approved for different types of cancer and different types of hereditary diseases. Cell therapy introduce healthy cell to repair or replace diseased tissue or cell, includes transplanting stem cell, immune cell, or genetically engineered cell. It is used to treat cancer. Gene therapy is also used to treat cancer. Cell therapy is also used to treat cancer. But cell therapy is used to treat autoimmune disorders, neurological disorders, diseases like rheumatoid arthritis. The cell is killing its own cell. CAR-T cell therapy, patient T cell are engineered to target their cancer cell for the treatment of certain blood cancer. Other cell engineered methods are also being explored. CRISPR gene editing method is used in this. Now, CRISPR's Cas and system, it enables precise editing of DNA sequencing. We are doing the editing of DNA sequencing. It has exciting potential application for correcting genetic diseases. So, different types of genetic diseases will be corrected with the help of CRISPR Cas system because it is doing the editing of DNA sequences. Gene therapy and cell therapy, there is a difference. It's referred to the process with cell therapy where the targeted cell are removed. We are removing the targeted cell and altered outside of the patient body. This involves the transfer of cell with the relevant function into the patient. In gene therapy, it refers to the removal or replacement in genetic material. Gene therapy, we are doing the changes in the genetic material, that is a DNA or RNA. This involves the transfer of genetic material, usually in carrier or vector in which with the help of vector, and the uptake of gene into the appropriate cell of the body. And cell therapy refers to the process where the targeted cell are removed. There is no need of any nucleic acid, DNA or RNA. There is no role of this cell therapy. As the name indicates, cell therapy. So removing the targeted cell are removed and altered outside of the patient body. This involved the transfer of cell with the relevant function into the patient. So gene therapy and cell therapy are both cutting edge medical approaches aimed at revolutionize healthcare. Gene therapy involves the manipulation introduction of genetic material, that is a DNA and RNA, into a patient cell to correct genetic disorder and enhance therapeutic effects. This can be achieved by delivering healthy genes to replace faulty one or modifying existing genes to improve their function. Cell therapy, on the other hand, utilizes the unique regenerative properties of stem cell or other functional cell types to repair or replace damaged tissue or organ. The other method is a CRISPR Cas9 system. It is a very powerful gene editing technology. We are doing the editing of gene, that DNA sequence, we are doing the editing that function like a pair of molecular seizure to cut DNA at a specific location. So in this method, we are doing the gene editing and we are cutting the DNA at a specific location with the help of restriction enzyme, molecular seizure, which allows scientists to modify genes. In this method, we are modifying the genes, we are editing the gene, we are cutting the gene. So it consists of two main components, CAS and an enzyme that cleaves DNA. CAS, the enzyme we are which we are using in this technology is not restriction enzyme. We are using a CASN, an enzyme that cleaves DNA, and a guide RNA that direct the Cas9 enzyme to the target DNA sequence. This system was originally discovered as an adaptive immune defense in bacteria. Adaptive immune defense in bacteria. So in this case, there is a given here. So the enzyme which is used is Cas9 enzymes, which help in the cutting of the cleaves DNA, cleaving the DNA, cutting the DNA, then doing the gene editing in the DNA sequence. Application, what is the application in this? Application is this, that it allows scientists to precisely edit genes in various organisms. It helps use in research to study. It is helpful in research field also to study gene function and develop new therapies. There is a great potential for CRISPR Cas9 to treat genetic diseases and other medical conditions. So these are the benefit of this technology. Now, regenerative medicine. Regenerative medicine is also important for in the field of biotechnology. It give rise to tissue engineering, which creates scaffolds and cell cultures to regenerate damaged tissue or organs. This involves stem cell, biomaterials, bioreactors, mechanical stimulation, 3D bioprinting, which we have learned in the previous slides. So these are the biomaterials. Bioengineered skin substitute growth factors and skill cell sprays are used to promote rapid healing of wounds with minimal scarring. So bioengineered skin substitutes, growth factors, and skin cell spray are used to promote rapid healing of wounds. Wound healing, it helps in the wound healing with minimal scarring. It also help in the biotechnology, also help in the development of artificial organs, devices that replicate the physiological function of organs, which we have learned in during the 3D printed organs. They're used to temporarily replace organ function in critical patient as a bridge to transplants. It helps in the formation of bionic organs, advanced artificial organs, that is known as a bionic organs. It may incorporate tissue derived from stem cell and biological molecules to better mimic natural organs. Some also have robotic capabilities, robot-like capabilities. It also helps in the development of bioartificial organs. This consists of biological components like cells and tissues integrated with artificial components that aim to provide biologically functionality akin to real organs, function like a real organs. These are the medical devices and the function biosensor, lab on a chip, microarrays, nanomedicine. So biosensor devices use biological elements. It helps to use for medical diagnosis, glucose monitoring, for the diabetes patient, pathogen detection, drug development. Lab on a chip, it's a microfluidic chips which help to diagnose different types of disease, drug screening, detection of toxin or pathogen, microarrays, which is the different types of biological probes to detect multiple biomarkers. It is used for diagnosis, personalized medicine, and disease surveillance. Nanomedicine, it is using nanoparticles, which is helping diagnosis different types of diseases, bioimaging, different types of therapies it is able to give, and generates regenerative medicine. So these are the application and these are the medical devices. Application of biotechnology research and developments. In research and development, there is a development in the field of genomics and proteomics and biosensor and diagnostics. So genomics and proteomics, it revolutionized research and development across diverse domains. In genomics and proteomics, tools like DNA sequencing unravel genetic complexity. Drug discovery benefits from recombinant DNA technology, expediting therapeutic advancement. Stem cell research explores regenerative potential. Industrial R&D optimizes bioprocessing for large-scale production. It helps in the vaccine development, biotechnological platform for safer immunization. So these are the field associated with the genomics and proteomics. Now, help in the develop biotechnology, help in the research and development, it helps in the development of biosensor and diagnostic also. With we have learned that it helps in the enzyme engineering, transform industrial process. While CRISPR technology, it facilitates precise genetic editing, which we have learned in the previous slide. It also help in multi-dimensional role in R&D underscore its pivotal contribution to scientific discovery, medical breakthroughs, and technological innovation. So it plays a pivotal roles in technological innovation as well. There is also with the help of biotechnology, there is a technology name is omics biotechnology, which we have learned genomics, proteomics, metabolomics, which understands cellular biology and disease mechanism. Metabolomics means biology and disease mechanism. Microbiome analysis, with the help of which we are provides insight into different types of health and disease, and using the field that is a bioinformatics. So biotechnology, application of biotechnology in transgenic animals. It's a very important field also. With the help of this, we are developing the transgenic animals, which is genetically manipulated to express foreign genes. With the help of this, we are genetically manipulated to express foreign gene. In July 2000, researchers from the team that produced Dolly ship reports success in producing transgenic lambs in which the transgene has been inserted at a specific site in the genome and function well. So application of biotechnology in transgenic animals, it serves diverse purposes, such as with the help of this, we can study different type of gene regulation, modeling human diseases for research, producing biological products like human proteins, testing the safety of vaccine and chemicals. So this is the reason we are using the transgenic animals to test the safety of different types of vaccine and chemicals, to develop the different types of proteins, to know about the different types of human diseases. So we are using these animals for transgenic purpose. So transgenic animal is one that carries a foreign gene that has been deliberately inserted into its genome. The foreign gene is constructed using with the help of technology. The technology is genetic engineering technology, recombinant DNA methodology. So in addition to the gene itself, the DNA usually includes other sequences to enable it to be incorporated into the DNA of the host and to be expressed correctly by the cell of the host. Transgenic sheep and goats have been produced that express foreign proteins in their milk. So in this way, we have improved the quality of milk, production of milk. Transgenic chickens also develop, which are now able to synthesize human proteins in the white of their eggs. These animals should eventually prove to be valuable sources of protein for human therapy. So these are the application of biotechnology in transgenic animals. With the help of three methodologies, we are developing the transgenic animals. First one, the embryonic stem cell, and second one is the pronuclei method. Embryonic stem cell select for cell expressing desired gene, then inject transformed embryonic stem into inner cell mass, and then after implant in uterus, and then test offering for presence of gene. Next one, the method is a pronuclei method, where desired gene is directly inserted into the vector, then implant in the uterus. Test offspring for presence of gene. This is a foster mother, and these give rise to desired products. So in like this, with the help of recombinant DNA technology method, we are developing the transgenic animals, transgenic organisms, which are important for model organism to study different types of diseases like Alzheimer's disease, heart disease, diabetes. It also help help researchers to understand the role of specific genes in development diseases and normal physiological process. It also help to test potential drugs and therapies. It also help in development of different types of vaccine. Example of biotechnology in health. Health, the most important thing is the genetically engineered insulin. Management of adult onset diabetes requires regular insulin because the rate of diabetes is increasing across the world. Earlier, insulin from cattle or pigs caused allergies, but genetically engineered insulin offers a safer alternative. So we are developing the insulin. That insulin is known as a recombinant insulin, which is a type of insulin that is produced using DNA technology. It is made by inserting the gene that codes for insulin into a bacteria or yeast cell, which then produces the human protein. So bacterial plasmids and human insulin gene is taken, then section of human DNA is taken with the help of restriction enzyme. Bacterial plasmid is taken, and then it is inserted into the plasmid, ligated, joined with the help of ligase enzyme, inserted into the host bacteria with insulin gene grown in fermenter, and then afterward insulin is separated and purified. So in this way, recombinant DNA technology, we are producing the human insulin. The ethics of genetically engineering. So what we have discussed, the bacteria has a own chromosome and other extra chromosome that is known as the plasmids. Plasmid is double membrane extra chromosome which is able to circular DNA, which is able to self-replicate, self-duplicate. So in its self-duplicate. So in the field of biotechnology, we are using plasmids or different types as a vector, and cosmid, bacteriophage, different types of vectors we are using as a vehicle to carry the alien piece of genes in the field of biotechnology. The ethics of genetic engineering is to it cures for diseases such as cystic fibrosis and cancer. Food which is healthier, stay fresh for longer periods. Risk is we are doing the recombinant DNA technology. There is a risk also new dangerous diseases being created. It is against God and nature. This is the ethical issues coming. Why there is a need for preparing genetically engineered insulins. In person with diabetes, the pancreas may not create enough insulin or it may not operate effectively, resulting in excessive blood sugar level. Diabetes patients now have an alternate source of insulin thanks to genetically engineered modified insulin, which is discovered by the with the help of recombinant DNA technology. Prior to the development of genetically modified insulin, insulin was taken from animal pancreas such as pigs and cows. This increased the possibility of contamination because we are taking from the pigs and cows. So there is a reason there was lot of chance of contamination with animals, viruses, and other pathogens. Because of genetically engineered insulin is designed to closely mimic human insulin, it is more successful than animal insulin in managing blood sugar level for the diabetes patient. Genetically modified insulin is readily available and can be manufactured in massive amounts. Now, day by day, the diabetes patient is increasing. Genetic engineering enables insulin to be tailored to the specific demands of diabetic patients. Several varieties of insulin, for example, can be manufactured with various rates of absorption and duration of action. Now about the genetically engineered insulin. Genetic engineered insulin is produced by inserting the human insulin gene into bacteria or yeast cell using recombinant DNA technology. So we are producing the genetically engineered insulin with the help of the technology is a recombinant DNA technology, and we are inserting the gene, the human insulin gene into bacteria or yeast cell, inserting with the help of vector. First, to know about the insulin. Insulin is synthesized by the beta cell of the islets of Langerhans in the pancreas. It controls the level of glucose in the blood, in the human's blood. As insulin deficiency manifests itself as a diabetes mellitus, whose symptoms can be alleviated by continuing program of insulin injection, thereby supplementing the limited amount of hormone synthesized by the patient's pancreas. Insulin used for diabetes was earlier extracted from where? We are extracting from pancreas of slaughtered cattle and pigs. So animal insulin is generally satisfactory through though cause allergy in some patients. So we are taking the earlier we are slaughtering taking the we are taking from the slaughtered peaks and which cause different types of allergies. So insulin cannot be orally administered to the diabetic patient because it degrades its alimentary canals. So we cannot take it orally because it degrades it was degrading alimentary gland of patients. So now we have developed the genetically engineering insulin with the help of human insulin, taking the human insulin gene and then incorporating into bacteria or yeast cell using the method is the recombinant DNA technology or genetic engineering. These cells are grown under the controlled condition to produce large amount of large quantities, which cheaper and easier to produce than animal driven insulin. We are the first generated insulin was developed by Dr. Herbert Boyer and Dr. Stanley Cohen in the year 1978. The first genetically engineered insulin was developed by, and it was approved by the United States Food and Drug FDA, Food and Drug Administration in the year 1988 after human trials. Since then, genetically engineered insulin has become the standard treatment for diabetes worldwide. It was helping millions of people to manage their condition effectively and lead everyday lives. So it is helping right now also. Previously, insulin we are taking from the extracting from the pancreas of slaughtered cattle and pigs. However, animal driven insulin contain different types of foreign proteins that caused allergies or other adverse reaction in some patients. Now, insulin comprises two short polypeptide chains, chain A and chain B. Insulin comprises two short about the, we move to the, we are making the recombinant DNA technology. We are taking the human insulin. Okay, human DNA. So it consists of two polypeptide chain A and chain B. About the difference between the previously the insulin we were taking from the slaughtered uh, pig and then recombinant insulin we are developing right now. So we are previously we are taking the from the slaughtered cattle and pigs. It is extracted from the pancreas of animals, contains impurities. Some people face allergies, limited availability. Only types are available, regular and NPH. Dosage varies based on the animal source and batch. Now, what we have developed recombinant insulin, that is a humulin. It produced using genetic engineering. It produced in the laboratory, highly purified, low risk of allergy, widely available, includes rapid acting, long acting, and combination formulation, more consistent doses due to precise amount. So in 19 in the year now it's about the again in the history about the development of the human insulin. In 1983, the American company Eli Lilly overcome this challenge, and this company prepared the two DNA sequence corresponding to human insulin A and B chains. It consists of two polypeptide A and B chains. This DNA sequence were introduced into plasmid E. coli to produce insulin chains A and B and separately. The chains were extracted and combined by creating disulfide bonds to form mature human insulin. So this is a method we have to produce the human insulin. Human cell with genes for healthy insulin are selected. So human cell with gene for healthy insulin are selected. Then a chromosome is removed from the cell, and from there we will take the allele piece gene of interest or gene which is responsible for controlling formation of insulin. Then we take the bacteria plasmid, we will take it, and then we isolate the plasmid, cut the plasmid with the restriction enzyme, and then cut the gene which is responsible from the human cell, which is responsible for the formation of insulin. Then insert into the gene inserted in plasmid with the help of ligase enzymes, and then it is inserted in the plasmid with genes inserted bacteria, and the human insulin is inserted into the plasmids with ligase enzymes. The plasmid is returned back to the bacterial cell. The bacterial cell allowed to reproduce. The bacterial cell allowed to reproduce in a fermenter. All the cell produce contain plasmids, and these all the cell become a recombinant plasmids which contain the human insulin gene. This is the process of recombinant insulin production with the help of genetic engineering techniques. So in mammals, including human, insulin is produced as a prohormone. In mammals, including humans, insulin is synthesized as a prohormone. Prohormone means like a pro-enzyme. The prohormone also needs to be processed before it becomes a fully matured and functional hormone. So prohormone, we have to process it before it becomes a fully mature and functional hormone. And the gene for this protein synthesis is located on chromosome 11. Where it is located? It is located on chromosome 11. This prohormone which contains a extra stage called the C-peptide. This prohormone which contains the extra stage is known as a C-peptide. This C-peptide is not present in the mature insulin and is removed during maturation into insulin. The main challenge for producing of insulin using recombinant DNA technology was getting insulin assembled into a mature form. So this one has been developed by the Eli Lilly, an American company, which prepared two DNA sequences corresponding to A and B. And what the company has done is they prepared the two DNA sequence corresponding to A and B chains of human insulin and introduced them in plasmids. Plasmid name is a PBR322, and then transported in the bacteria. The bacteria name is the previously which I explained. The bacteria name is E. coli, and then inserted into the bacteria. Sequences for A and B chains were linked with LAGZ gene and introduced into PBR322. The transformed E. coli could be selected by blue-white screening method. So there is a, when it is developed, it is selected with the help of screening method. The screening method is blue-white screening method. Chains A and B were produced separately. We have produced chain A and B. Chain A and B is produced separately, then extracted and combined by creating disulfide bonds to form human insulin. So we have removed the extra C peptide. So this is the method. It is given here. The importance of this process because it is acceptable to people with a range of religious belief who may not be allowed to use insulin from animals such as cows or pigs. The product, the human insulin, humulin is very pure. Human insulin can be made on a commercial scale and it reduces cost. Now, next one about the gene therapy. Gene therapy is attempt to correct the hereditary disease or genetic disease in any person. It is a collection of method that allows correction of gene defect that has been diagnosed in a child and or embryo. So we are diagnosing it in a child or embryo. Here genes are inserted into a person's cell and tissues to treat a disease. In this method, we are connecting a defective genes involving delivery of normal gene into the individual or embryo to take over the function of and compensate for the non-functional genes. So it is an important technique to prevent different types of diseases, to cure different types of diseases. It involves adding new copies of genes or replacing defective genes with a healthy one. So we are removing the defective gene and inserting the healthy genes. So it can be used to address both inherited genetic diseases like hemophilia and sickle cell disease and acquired disorders like leukemia, like blood cancer. So there are different approaches in that: gene addition, gene editing in vivo and ex vivo. Gene addition, we are introducing a new gene to help the body fight diseases. Gene editing, as the name suggests, we are doing the targeted changes to specific DNA sequence. Gene editing in vivo and ex vivo. In vivo into the body, ex vivo outside the body in the laboratory. So these are the method of the gene therapy. So gene therapy is a collection of methods, correction of gene defect that has been diagnosed in child or embryo. The first clinical gene therapy was given in the year 1990 to a 4-year-old girl with adenosine deaminase deficiency, ADA deficiency. This enzyme is crucial for the immune system to function because in its absence, lymphocyte proliferation is inhibited. Without T cell, ADA deficient children are wide open to the different types of attacks of diseases and viruses. So the child suffer from severe combined immunodeficiency disorder, which is caused due to deletion of gene for adenosine deaminase. So the first clinical gene therapy was given in the year 1990 to a 4-year child girl, which is suffering from the ADA deficiency, adenosine deaminase deficiency. The child is not having the adenosine deaminase enzymes, which is responsible for the proper functioning of the T cell. Lymphocyte so this is the reason in its absence lymphocyte proliferation is inhibited, and the child suffer from different types of severe combined immunodeficiency disorder, SCID, is because of the deletion of gene for adenosine deaminase. So in some children, ADA deficiency, adenosine deaminase deficiency, can be treated by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy. So these are the method. So there are types of somatic cell therapy. We are doing the somatic cell body cell, which will not be inherited later generation. When we are doing the treatment in germ line gene therapy, we are doing in the germ cell, that is the of the eggs and sperm. So in this case, it is heritable because it is doing in the germ cell eggs and sperm. So but for the reason, for safety, ethical and technical reason, it is not being attempted at present. So we are not using the germline gene therapy because it is going incorporated into the eggs and sperms. Ex vivo and in vivo is ex vivo outside the body. In vivo is inside the body. Specific cell type is T cell, target, and stem cell. And in vivo, tissue specific T cell. So gene delivery method is given, and then control over process, stability, and examples are different types of diseases which we are treating in this. So these are the two types of gene therapies. Example of gene therapy application is it treats different type of genetic diseases like hemophilia, sickle cell disease, cystic fibrosis, spinal muscular atrophy. It's help in the treatment of leukemia, lymphoma, blood cancer like that, and lymphoma, lymph cancer. Other diseases, gene therapy is also being investigated for condition like AIDS, diabetes, and heart diseases. So for the treatment, what we are doing as first step towards gene therapy, lymphocyte we are taking from the blood of the patients. So treatment, what we are doing, lymphocyte from the blood of the patient are grown in culture outside the body. A functional aDNA, that is known as a cDNA, using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolated from bone marrow producing ADA is introduced into the cell at early embryonic stage, it could be a permanent cure. So, if a person is suffering from the ADA deficiency, that is a adenosine deaminase deficiency. So, if the gene is isolated from bone marrow, producing ADA is introduced into cell at early embryonic stage, it could be a permanent cure. Gene therapy to cure sickle cell anemia. So extract stem cell from patient bone marrow, infuse stem cell with normal gene for hemoglobin, condition patient's bone marrow to receive new stem cell, insert treated stem cell into patient. So these are the treatment of benefits of gene therapy. Target the root cause, potential for a cure. It cure different types of genetic diseases. We have to, it has the benefits to provide personalized medicine, individual based on the individual genetic makeup. But there are some challenges also in the field of gene therapy. Immune response, the body may react negatively to the introduced, what we are introducing the gene of vectors. There may be a chances that there may be a negative immune response. The new gene might insert into the wrong place in the genome. There may be a chances. So off target effects also, long term effect also are still being studied. Accessibility and cost, it is a little bit expensive or more expensive, you can say, and may not be accessible to everyone. FDA has approved several gene therapy for use in humans, including for the diseases. Diseases are cancer, spinal muscular atrophy, hemophilia, and sickle cell disease. Now about the molecular diagnosis, how we are diagnosis the molecular diagnostic tool, which is a very important tool in the laboratory which we develop, is the ELISA. ELISA is the enzyme-linked immunosorbent assay test that detects and measure antibodies in your blood. So ELISA is specifically we are doing for the AIDS patient, acquired immunodeficiency syndrome patient. It is caused by the retrovirus and the name is the HIV. It was also for Zika virus and other diseases. So, and ELISA is and ELISA treatment is very important in the field of molecular biology. Now, about the what is antibody? Antibody is a protein that our body produce in response to harmful substances. The antigen, which is the outside substances, and antibody are produced that our body is making to fight against it. Advantages of biotechnology application in health. There are many application, very advantages, improved diagnostic, targeted therapies, vaccine developments, sustainable treatment, enhanced drug production, nutritional improvement, gene therapy, environmental impact reduction, research advancement, global health solution. So in this way, it provides affordable and accessible healthcare solution addressing health disparities and improving public health worldwide. There are few advantages also, disadvantages also. Every coin has a pros and cons. So in the field of biotechnology, there is some disadvantages. Now, what are the disadvantages? Ethical concern, allergic reaction, high cost, accessibility issues, regulatory challenges, potential for resistance, environmental impact. Genetically modified organism may have unforeseen effects on ecosystem, including biodiversity loss and disruption of natural habitats. Dependency on technology, we are totally relying on biotechnological solution, which may diminish traditional medical practices and knowledge. Uncertain long-term effects of gene therapies and genetically modified foods are not fully understood, raising concern and about safety and efficacy. So it raises social inequality. It focuses on biotechnological advancement might lead to neglect of other essential health determinants such as socioeconomic factors and access to basic healthcare services. So in next about the application of biotechnology is application in bioenergy, in creation of biofuels that are derived from biomass, and biomass are renewable and sustainable energy which we can replenish against. It's with a potential to replace fossil fuels. So there are, with the help of biofuels, there are different types of application which is increase biomass. It increases biomass per hectares, increase content of oils, biodiesel crops, fermentable sugars, ethanol crops, improved processing characteristic that facilitate their conversion to biofuels. Significance it is found to be useful for sustainable bioenergy synthesis via completing the wastewater treatment processes with electric energy synthesis. So these are the application. In conclusion, the application of biotechnology in health represents a transformative force in modern medicine, driving innovation that improve diagnostic, treatment, and patient outcomes. From personalized medicine and advanced therapies to sustainable practices and global collaboration, biotechnology holds the potential to address pressing health challenges and enhance the quality of life. As the research and technology continue to evolve, the integration of biotechnology into healthcare will pave the way for more effective, accessible, and affordable solutions, ultimately leading to a healthier future for individuals and community worldwide. So biotechnology is a transformative force in medicine, in health, offering new hope for treating and preventing different types of diseases, improving patient outcomes, patient life, and advancing the frontiers of healthcare. As the field continues to evolve, it promises to reshape the future of medicine and improve human health.